Hi everyone, welcome to my A-level biology lecture series. In this lesson, we will explore the regulations of gene expression in prokaryotes by using lag operons. At the end of this lesson, you will be able to describe the differences between structural genes and regulatory genes and the differences between repressible enzyme and inducible enzyme, and also explain genetic control of protein production in prokaryotes using the lag operons. Let's get started. Prokaryotes such as bacteria adapt to environmental changes by regulating gene expressions at the level of transcriptions. So many genes in the bacterial genome can be switched on, or we say is activated, or switched off, basically means deactivated in response to the changes in the metabolic status of the cell, which often triggered by environmental factors. So this regulation enables cells to control the production of enzyme by adjusting the transcriptions of the gene that encodes them. So at the transcriptional level, control involves regulating the synthesis of messenger RNA, which carry the genetic code for this enzyme. The fundamental mechanism for transcriptional regulations in bacteria is the operons model, where a group of genes, or commonly known as the structural genes, is regulated together under the control of a single promoter. One of the examples that we are going to use it to study how this gene can be switched on and switched off is the lag operon. Lag operon is an inducible operon which is activated in the presence of lactose. So generally, when lactose is absent, this operon or lag operon is deactivated. Another operon that we are going to look at it, but it is not in our syllabus, is the tryptophan operon. Tryptophan operon is a repressible operon. Bacteria will switch it off when tryptophan is abundant. Let us look at the lactose operon, or commonly known as the lag operon, which is an inducible operon in prokaryotic gene regulation. So the lag operon is an example of an inducible operon in E. coli that enable this E. coli to metabolize lactose, provided that lactose is present in the environment. So the component of the lag operons are promoter, operator, and 3 structural genes. So you can see that in this diagram, lag operon, consists of promoter, operator, and 3 structural genes. They are lag Z, lag Y, and lag A. At this moment, operator is bound with the repressor protein produced or coded by repressor gene or regulatory gene. So what is the functions of a promoter? So a promoter is a specific DNA sequence where RNA polymerase bind to initiate transcription. So in the lag operon, a single promoter controls transcriptions of all 3 structural genes. How about operator? So operator acts as a control switch, or molecular switch in this case. So it is a binding site for an active repressor protein. When an active repressor protein binds to it, RNA polymerase cannot transcribe the structural gene. So what other structural genes can be found in lag operons? So the gene code for polypeptide or enzyme directly involved in the cell structure or metabolism are known as structural genes. please take note that structural genes does not code for structural protein, because students always confuse about the structural gene and structural protein. So please bear in mind that structural genes are the gene code for polypeptide or enzyme directly involved in the cell structure or cell's metabolism. So the structural genes of the lag operons are lag Z, which code for beta-galactosidase, lag Y code for permease, and lag A code for trans-acetylase. Beta-galactosidase hydrolyzes lactose into glucose and galactose. Permease, again from the word permease, permeability, basically facilitates the lactose uptake into the cells. And trans-acetylase transfer an acetyl group from acetyl-CoA to beta-galactoside. So in this case, we can see that these three genes are involved in the lactose metabolism. So it makes sense that without lactose, these three genes are deactivated or inactivated due to the lag operon inactivation. So what is a regulatory gene? So this regulatory gene is located near to the lag operon. So it is transcribed into mRNA, and this mRNA is translated into a repressor protein. And this repressor protein is active and it binds to the operator, preventing RNA polymerase from attaching to the promoter. So therefore, this blocks the transcriptions of the structural genes and therefore keep the operon switched off in the absence of lactose. So we can see that in this diagram, this operator, can you see that? Operator is bound by this active repressor, and this active repressor is synthesized or coded by a regulatory gene here. So this active repressor, when they bind to the operator, RNA polymerase can't bind to the promoter. Therefore, there is no transcriptions of the structural genes. So let us look at the lag operon's mechanism in the presence and absence of lactose. So by looking into this diagram, so just now we have learned about how these repressor protein bind to the operator. But now with the in the presence of the allolactose, where the lactose is converted to allolactose, allolactose can bind to the allosteric sites of this repressor protein, making this repressor protein to become inactive. So inactive repressor protein detaches from the operator. Therefore, RNA polymerase can bind to the promoter and initiates the transcription, results in the productions of the mRNA. So the translation of this mRNA produces beta-galactosidase, permease, and trans-acetylase, and these three enzymes are involved in the lactose metabolism. So therefore, we can actually imagine this. When lactose is available, the cells need to metabolize lactose. So therefore, the operon is switched on. However, in the absence of the lactose, there is no point for the bacteria to transcribe these three structural genes because there is no necessity to produce the enzyme to metabolize lactose. So in the absence of lactose, the regulatory gene is transcribed into the mRNA molecule that is translated into an active repressor. So the repressor binds to the operator, blocking RNA polymerase from binding to the promoter. And with RNA polymerase blocked, there is no transcriptions of the enzyme. So this prevents production of unnecessary enzyme and saving ATP when lactose is absent. However, in the presence of the lactose, lactose is converted into allolactose. And allolactose binds to the repressor, inactivating it, and therefore detach, or this inactive repressor protein detaches from the operator. So RNA polymerase can now bind to the promoter, and the transcriptions of the structural genes like Z, like Y, and like A begins. So the resulting mRNA is translated into beta-galactosidase, permease, and transacetylase. And these three enzymes enable the cells to transport and break down lactose for respiration. So now we can imagine, or we can actually figure out that, yep, this bacteria actually detect the stimuli from the surrounding. When the surrounding, when lactose is absent, therefore there is no requirement or no necessity for the cell to produce the enzyme that metabolize lactose. However, in the presence of lactose, it makes sense that for this bacteria to produce the enzyme which are involved in the breakdown of the lactose for respiration. And with that, we have come to the end of this lesson. Hope you found it helpful and easy to follow. If you have any question, feel free to drop them in the comments section. See you in the next lesson. Have a great day.